you. Thank you very much for the invitation to come speak. I'm very pleased to be here. This is my first visit at Copenhagen, and I've enjoyed the city immensely. It's beautiful here. Really nice visit. So I was asked to talk about amino acid regulation of glucagon secretion, and unlike Anders, I think that's a little bit of a big task to bite off, because amino acids are actually not just one species, right? Glucose is one molecule, and when we think about amino acids, that's really encompassing a lot of different molecules, and they all act by different mechanisms, we think, and perhaps converge on the regulation of glucagon secretion. But some of my work, I've been very interested in understanding how amino acids not only regulate glucagon secretion, but also determine alpha cell mass, and I think that's an important study that we'll talk about both. So of course, this idea that amino acids and hypoglycemia can stimulate, or low glucose, we'll say, can stimulate glucagon secretion is not new. We've known about the ability of amino acids to stimulate glucagon secretion for 60, almost 70 years. But the mechanisms by which they do this is still not clear, and so we'll talk a little bit about that. So of course, glucagon can stimulate hepatic glucose output from the liver, and that's one of the reasons why it's been quite popular for study for diabetic drugs. But I became very interested in how amino acids might regulate alpha cells in my postdoc, and put out this paper as a primary role for alpha cells as amino acid sensors. So I think this is a really important role for alpha cells, is to both sense amino acids and communicate through glucagon the disposal of amino acids through the liver. And so here are some data that first illustrate this. So this is, again, since we have a historical perspective, these are data from perfused rat pancreas, looking at the effects of arginine on glucagon secretion at low glucose. And I think what you can appreciate is this biphasic stimulation of glucagon secretion at increasing doses of arginine. And what I think you can also appreciate here is at low glucose, the effects on insulin are quite slow and tonic. So wait a second, amino acids stimulate both glucagon and insulin secretion. Well, that's true, but maybe they do so by different mechanisms, and then I'm also going to push this idea moving forward as well. And so if I do these same experiments, or look at the same data from, say, normal glucose, you'll see that there's a mild suppression of glucagon secretion in those rats, but it's not a complete loss. So again, amino acids are potent stimulators of glucagon secretion, even in the suppressive tone that's coming from glucose. So do all amino acids work the same? Well, we talked about data from multiple groups who've looked at amino acids' ability to stimulate glucagon secretion, and this is some newer data from our host here, looking at individual amino acids and their ability to stimulate glucagon secretion. So in this study, this is perfused mouse pancreas, and looking at individual amino acids at 10 millimolar of the amino acids, and you can see that each individual amino acid has slightly different potency at stimulating glucagon secretion. In fact, some of them stimulate very well, like cysteine, alanine, and some are a little less impressive, like glutamine, and so we'll talk about that as well. So thinking about how we might move these glucagon as a target for treating diabetes, Dan mentioned that blocking glucagon receptor signaling, while it does lower blood glucose, and it's very effective at that, has some off-target effects. And so I'll talk a little bit about some of our work, including some of our collaborative work with Dan, looking at global glucagon receptor knockout mice as well as liver-specific glucagon receptor knockout mice. So of course, while you don't achieve hypoglycemia, you do get a modest reduction in glycemia in mice that have globally lost the glucagon receptor, as well as an improvement in glucose tolerance. This is an IPGTT. But what you also get, so if you use a small molecule like a monoclonal antibody against the glucagon receptor, you get a lowering blood glucose, but you also get this concomitant increase in glucagon. So you get hyperglucagonemia. That's not really an attractive thing. And so we became very interested in the mechanisms of how this might occur. So of course you have on the left a normal pancreas with amylase in red and glucagon in green. And then on the right you have this glucagon receptor knockout mouse where you have about a 13-fold increase in alpha cell mass by 5 months of age. So this is a really dramatic increase. And by the time they're 12 months of age, I can resect out little tumors out of these mice with almost my naked eye. Don't need a lot of magnification. So it's a pretty impressive phenotype. And we became very interested in understanding if the mechanism of how this happened. So we then also asked if this happens for humans. So we performed experiments where we transplanted human islets under the kidney capsule of mice, and we treated those mice with a glucagon receptor monoclonal antibody. And after a few weeks, we measured the glucagon content in those islets as well as their proliferation of those alpha cells using Ki67 as a marker, which you'll see quite a bit. And you can see that there's an increase in glucagon content in alpha cells. These are human islets, right? So a lot of things that we think of that cause beta cell growth in mouse don't really translate over to the human. This is actually something that really potently stimulates alpha cell growth in human islets. So I think that's pretty remarkable. So how does it occur? So I took this approach of looking at glucagon receptor knockout mice as well as monoclonal antibody treated mice, and we were pretty sure that the signal must be coming from liver based on some of our work with the liver-specific glucagon receptor knockout mice that Dan's lab had made. And we looked at a lot of different peptides that might be changing because we were very convinced this must be some sort of small molecule or peptide. And when we tried all these different changes that we saw in serum levels of different proteins, none of them stimulated alpha cell proliferation in an ex vivo model. So the really glaring, obvious answer was actually in the effects of glucagon on amino acid transport and catabolism. So we know that glucagon is responsible for the fractional extraction of amino acids from blood. And so it made a lot of sense that if you block glucagon receptor signaling and block this process, you would develop hyperaminoacidemia, and that's exactly what you see. So it's kind of busy, but I think you can appreciate a couple things. One, amino acid levels circulate at different concentrations across amino acid species. So for example, aspartate is much lower than glutamine, so glutamine's up around 500 or 600 500 or 600 micromolar, under normal conditions. This is shown here with a white bar. And if you block glucagon signaling, you get an increase in most amino acids, including glutamine, coming up here around 3 millimolar, as well as arginine. But most amino acids change. But I'm going to focus on these two for the sake of our talk, because I think they're really important at determining alpha cell mass, but maybe they have some divergent roles on secretion. So to really test this idea that amino acids could potently regulate alpha cell growth, we made media that recapitulated the serum amino acid levels that we observed in mice. So we made media that had wild-type serum amino acid levels, called the low amino acid here, and we made media that has the glucagon receptor serum levels, shown here with high amino acids. And you can see there's a potent stimulation in alpha cell proliferation. This is at 3 days. We didn't see an increase in beta cells and delta cells. That may not be the case as you treat for longer, and so we could talk about that in the discussion if you're interested. But we think that this is due to the ability of alpha cells to have a very strong sensing mechanism for amino acids. So what amino acids are important? We showed that glutamine was an important driver of alpha cell growth. So here we have treated with increasing dosage of glutamine. We're keeping all other amino acids high, like we observed in the glucagon receptor knockout serum. And once you pass this threshold between 500 and 2 millimolar, 0.5 and 2 millimolar glutamine, you start to increase alpha cell proliferation. So this is the transition between what you might observe with a glucagon receptor antibody treatment and glucagon receptor knockout mouse. So these data, and lots of other people have worked on this, and this is actually one of the things that I'm I'm really most proud of is that these data are actually really reproducible between groups. I think that there's been a tremendous consensus that the liver can regulate this fractional extraction of amino acids, which then in turn regulate alpha cell function. So we know it regulates glucagon secretion, but we also think that it also regulates alpha cell mass. And so how it might do that, I think, is of also interest. So we showed you that glutamine is required, but we don't know which other amino acids are regulated alpha cell mass. And so kind of to start to get at that, I'm going to show these data again from the 1970s, where I think this might be some of the first demonstration of the liver alpha cell axis at work, right? So this is a low glucose, again, rat perfusion, showing 20 millimolar glutamine and 20 millimolar arginine perfused into rats who had been maintained on either a controlled diet or a high protein diet, where you might expect amino acid circulating levels to be higher, right? Because they're getting increased dietary exposure to amino acids. And what you can appreciate is that there's actually this increase in glucagon secretion. And so we wonder if this is sort of a demonstration that maybe these mice had either perturbations in their ability to extract amino acids out of the blood, or if this is a signal that there was some sort of alpha cell hyperplasia or changes in alpha cell mass in these mice, or these rats, excuse me. But I think it's also important to note that it's not just protogenic amino acids that can exert their effects on alpha cells. So here we have ornithine as a potent stimulator of glucagon secretion as well. And if you get this high protein diet effect with certain amino acids. So we became interested in understanding, so I showed you that glutamine can regulate both glucagon secretion there. We became very interested in understanding how this might work. So we had previously shown that glutamine transporters expressed on alpha cells can be induced when you block glucagon receptor signaling. Interestingly, this happens in mouse, but maybe not so much in human, or at least the specific transporter we'd worked on at SLC3085 didn't happen in humans. So we're still not quite understanding how human cells might respond to these, but we still think it's driven by amino acid transporters, and then in some cases their subsequent metabolism. So to kind of get at this, we said, okay, well, we know glutamine stimulates both glucagon secretion as well as alpha cell increases in alpha cell mass. How does it do it? And so we first looked at the enzyme downstream of that mediates glutamine metabolism, that's the glutaminase. And so when we look at glutaminase gene expression, these are data from Mark Huesing's group, looking at alpha, beta, and delta cells from mice, and you can appreciate that glutaminase is highly expressed in alpha cells and maybe not as much in other islet cells, whereas the liver isoform of glutaminase, glutamase 2, is very low in all islet cells. We saw this by staining as well, so you can appreciate that here in red we have glutaminase 1 protein expression, and we are marking the glucagon positive cells in green, and you can see that there's increased expression of glutaminase 1 in mouse pancreas. We see that this is due to two different isoforms of glutaminase, so two different isoforms can be produced from glutaminase based off alternative splicing, so the KGA isoform, which is the kidney-derived, and then this GAC isoform, which we think is a mitochondrial glutaminase, which might have a really important role in thinking about how substrates are delivered to certain pathways in the TCA cycle. And so we see this heightened expression of GAC, and it looks like it has some punctate expression as well. So we're wondering if this is real evidence that in alpha cells, glutaminase is in the mitochondria. So we also see this in human pancreas. So again, you can see very little expression in the excrement compared to the islet, although there is quite a little bit more in human pancreas. But you can see that the glutaminase, the very intense expression colocalizes very nicely to alpha cells. We see this in type 1 diabetic pancreas as well as type 2. So it looks like this is conserved. So we began to ask, how does glutamine regulate alpha cell function and and as well as this growth. So we use this small molecule inhibitor that's currently in clinical trials for treating cancer, the CB839. And we found that you can dose-dependently inhibit alpha cell proliferation in response to hyperaminoacidemia in cultured mouse islets. Taking it a little step further, we began to look at the specificity of knocking out glutaminase and alpha cells alone. So here we've knocked out, using Klaus Kessner's glucagon Cree, we've knocked out the glutaminase gene selectively in alpha cells. And then we gave them an arginine bolus, and we were kind of surprised. Knocking out glutaminase really had little effect on glucagon, at least in circulating glucagon. And so I think this is still a little bit of an open question, as how glutamine might stimulate glucagon secretion. In fact, we find that this is an arginine bolus, you'll notice. We didn't stimulate with glutamine, right? And that's because we don't really get much of an effect with glutamine when we do these studies. So I think the jury is still out on how glutamine stimulates glucagon secretion. And I think it probably has a lot to do with how people set up their experiments, whether you do things in, say, RPMI or DMEM media that has 4 millimolar glutamine existing in the media versus, say, something like a Ringer's buffer. And you might get very different results. So what about proliferation? So I told you glutamine stimulates alpha cell proliferation, so we did these same experiments and treated the mice for 2 weeks with glucagon receptor monoclonal antibodies. So here, we did both controls. We did the Cree-positive mice because we were concerned about this knock-in into the glucagon locus, as well as we did the Cree-negative flox mice, and you can see that there's no difference in the alpha cell proliferation once you stimulate with the antibody. But if you knock out the glutaminase allele in alpha cells selectively, you can markedly reduce. So we know that, like the CB839 data, that glutaminase action is required for proliferation, maybe not as much for secretion. So we know that glutamine is important at driving this liver alpha cell axis, at least as far as mass goes. But we knew that there were also other transporters that would be really important as well. Other amino acids that would be very important as well. And so we did some experiments to try to identify other amino acids that were playing an important role in the liver alpha cell axis. And what we found is that one of the most potent amino acids that we found is actually arginine. So similar to the studies that I showed you earlier with glutamine, we actually did batched effects. So we batched amino acids together 3 or 4 at a time and looked at how they affected glucagon secretion. So what we did is we keep all amino acids high, even glutamine, and then lower down amino acid levels of individual amino acids batched together to levels that we observe in wild-type mice. So these aren't losing the amino acids altogether, we're just dropping them down to what's normal levels. And you can appreciate that when we knock down this RHGN, which had lower glycine, arginine, histidine, and asparagine levels, that we lost the proliferative stimulus of high amino acids altogether. And only when we restored arginine were we able to restore the proliferative effects of high amino acid media on alpha cell proliferation. So we thought this meant that arginine was also playing a really critical role in alpha cell proliferation. And so we thought, how does arginine get into alpha cells? How does it exert these effects? We'd looked at glutamine transporters and glutamine metabolic enzymes, so we wanted to look at arginine transporters as well. And what we were surprised to find is that in human, shown here on the left, alpha cells in dark red and the beta cells in light blue, you can appreciate that SLC7A2 is the dominant arginine transporter. These are all arginine transporters expressed in islet cells. It's the dominant transporter in human. It's expressed highly as well in mouse. Maybe there's a few other arginine transporters that can also transport arginine into alpha cells. But we thought this was really impressive, that this one transporter is like the highest expressed transporter in islets, and its highest expressed is in alpha cells. So we thought it would be doing something really important. And so we looked at actually GWAS data. We found SNPs that were associated with HbA1c and intronic regions in the SLC7A2 gene. These are data in collaboration with Mark Keller and Alan Addy at University of Wisconsin. So we think that there are transcription binding factor sites in this intronic region that are driving SLC7A2 expression. We don't really know where SLC7A2 is important. We know it's expressed in alpha cells. We know it's expressed a little bit lower in beta cells. And we also know it's expressed in liver, if you were paying attention to my earlier slide with the gene expression from liver. So the first thing we did was we took a global knockout approach. We said, okay, what happens if you knock out SLC7A2? And here are the GTT data. We were underwhelmed. There was no effect. So knocking out SLC7A2 had no effect on glycemia. And so this didn't really fit with the HbA1c data. And so we were like, well, okay, so maybe this has no effect on physiology, but we found a profound effect actually on glucagon secretion. So here on the left are serum glucagon levels from fasting mice, and you can see there's a not statistically significant drop in glucagon levels in the serum of fasting mice from knockout animals shown here. But if you give them an arginine bolus, they have essentially no effect, right? So knocking out this transporter completely abolished the effect of arginine on glucagon secretion. And you can really appreciate that here in this parafusion data, where you get a very nice stimulatory effect with arginine in wild-type animals shown here in blue, but there's absolutely no secretion in these SLC7A2, or at least changes in secretion in this SLC7A2 knockout mice, not even when you hit them with KCL. So there's no changes in the alpha cell mass. We've measured alpha cell mass. There's no difference in the glucagon content of these islets. And so this really begs, why would they have no effect on glycemia if you've really disrupted glucagon secretion? Well, that's because we also have disrupted insulin secretion. So even though SLC7A2 is expressed at lower levels in beta cells, we've also affected the ability of arginine to stimulate insulin from beta cells as well. Interestingly, this is an effect that we can observe even in the heterozygous mice, so just knocking out one copy. So that maybe really supports this idea of a dosage effect with SLC7A2 expression on mediating the effects of these islets on arginine. But you still have an intact KCL. So what about proliferation? Well, if we knock out SLC7A2 in mice and stimulate them with glucagon receptor monoclonal antibodies, you can see that we reduce alpha cell proliferation. So this shows that arginine is important for regulating alpha cell proliferation. We see a loss of mTOR activation. We've previously shown that this is an mTOR-dependent pathway. And so you lose your mTOR-dependent stimulation through the amino acid pathway. And then we also observed a loss of this SLC38A5 transporter, which I told you was a glutamine transporter. It transports other amino acids as well. But we lose the induction that occurs, which we know is an mTOR-dependent pathway, mTOR-dependent pathway in the knockout mice as well. And then finally, if we just take out their islets and culture them, we also see a loss of transporter expression. Or excuse me, you see a loss of proliferation as well. So I'll just summarize really quickly. One thing that I think is important, we don't really understand how these amino acids work. For example, is arginine acting through its cationic properties and affecting membrane potential? We don't really fully understand that yet. Based on some earlier work by William Lays and others, it looks like that's probably the case. Certainly when we start to target amino acid metabolism, we do see some increases in glucagon secretion that supports that. Data that I didn't have a chance to show you. But I want you to understand that there's also this intersection between these amino acid pathways as well. So here we have glutamine coming in, and intersecting here at the ornithine transferase, you have an intersection between arginine and glutamine. So just to summarize, arginine can stimulate glucagon secretion. Glutamine in our hands, not so much, but at least through some mTOR-dependent mechanism, we see some interaction between the arginine and glutamine pathways, and certainly we find that glutamine metabolism is required, at least for proliferation as well. So some caveats. Amino acids don't exist in isolation. They have multiple mechanisms of action. Oftentimes we do our experiments with really super physiological levels of amino acids, like 10 millimolar, which, I showed you the highest amino acids like alanine and glutamine at normal levels are sitting in the 500 to 600 or 600,000 micromolar range. So maybe there are other things that can explain why amino acids have such potent effects on glucagon secretion. And so I'll just put a plug for my buddy John Campbell and his work showing that maybe in mixed meal tolerance, some of the effects might be mediated through incretins on alpha cells. So I'll thank my long-term mentor, Al Powers and his group, as well as our funding and the people who did this work in my group.
Right. Thank you, Danielle. Questions? Timo.
Did you try to treat your SLC knockout islets with sulfonylurea to see whether you can still get the pathway when you...
I'd say two things that we really should have done. We didn't use any kind of sulfonylurea inhibitor, and the other thing we didn't really test is their ability to respond to cyclic AMP. I think these are two places that we really should go back and invest some of our time. I'm sure a reviewer, this...
It would tell you whether you still can depolarize the cell.
Exactly. But, the KCL was really surprising. We thought that it would have an effect, and it didn't.
I think we have in the back.
Yes, thanks for the talk. The KCL effect is indeed quite surprising. So does it mean that there's excitotic defects when you knock out arginine receptor? And why did you use 20 millimolar KCL instead of 70 that you can have a higher stimulation? And also, did you measure the intracellular calcium? That would be quite nice to see whether calcium also rises, whether excitosis.
So what I didn't mention, we didn't use a higher dose. We only used the one. We should go back and try other things. We'll do those in static cultures. But we did make individual, we made flox mice of this, and we crossed them now to Gcamps as well. And so we're able to, study the alpha cell effects on calcium as well. But we haven't done those studies, so we don't know if this is a exocytosis. So we have plans to look at EM and make sure, even though they have the same content, is there something wrong with the actual vesicles? We didn't get any clues from gene expression data. I didn't show those. We did RNA sequencing on these mice, and nothing really popped up as significant. So we're not really sure. But our prevailing hypothesis is that maybe this has something to do with the membrane polarity, because arginine's cationic, and so that's my best guess.
Right. Two more questions. Patrick down here, and then Philip afterwards.
I think you are absolutely right when you say that it's concentration dependent. I mean, you used very high concentrations of arginine, 10 millimolar, whereas the concentration you have in plasma is maybe 0.3. And I think, we know a lot of these transporters. We know exactly how they transport arginine and other charged amino acids. I think the obvious interpretation of the lack of effect on basal is that the concentration of amino acids is not high enough to be transported by these electrogenic transporters.
So there's two different types that are expressed in pancreas, and that's something we need to dive a little deeper in. But the KM for one of them is around 350 micromolar, so it's getting to the area where we are at with our knockout mice. But the other one is much higher. It's in the millimolar range, right? And you wouldn't expect concentrations to get that high under even our glucagon receptor knockout mouse conditions. So it doesn't seem as likely of a mechanism to me.
All right, last question from Philip.
So thank you for a very interesting talk. I'm a big fan of your cell metabolism paper from 17. One question, have you looked into the amino acid transporters in the livers of your animals with and without glucagon signaling?
As far as the actual amino acid transporter levels, we see downregulation of things like SLC7A2 and other even mitochondrial transporters. So that really supports the idea that there's this decrease in the fractional extraction from blood, leading to that hyperaminoacidemia we observe.
All right. Thank you, Danielle.